Hello, and welcome back to Mammalian Anatomy at Cuca College. Today what we're going to do is continue our lecture series on Chapter 16, the special senses. And in this video, we're going to focus on the anatomy of the eye. The eye is one of the organs that I just absolutely geek out about. I can't tell you how amazing and awe-inspiring the eye is because it just has a beautiful complexity and a beautiful order to it that I just think it's such an elegant organ. And I can't wait to tell you about its anatomy, and then we can go into like the optics and physics behind the eye. And it's just super, super cool. And so I can't wait to show you. Yeah, so let's dive right into it. So the eye is actually composed of three layers. You may hear me call them tunics because I use the old words for things sometimes. So there's a fibrous tunic, vascular tunic, and neural tunic or inner tunic. And your book uses the more modern names: fibrous layer, vascular layer, and inner layer. So on this diagram, you can actually see that there are going to be three layers. There's an outer layer here, a middle vascular layer there, and then an inner layer here. So let's go into these layers, see what they're composed of, and what roles they play in the function of the eye. So the outermost layer is going to be the fibrous layer, and it's made out of dense fibrous connective tissue. And so it's going to be located here and here. There's actually two regions of the fibrous layer. There's going to be the sclera. The sclera is going to be the whites of the eye, and it is very, very strong, and its function is to act as an attachment point for the muscles that we talked about in accessory structures. Also, it helps maintain the eye's overall shape. And we'll see later on when we talk about the optics of the eye, shape is essential for being able to focus light. Over here, there's a clear part of the fibrous layer. This clear part is called the cornea. The cornea is going to allow light to enter the eye, and also it has this quite a bit of a curvature to it. This curvature is essential for refracting light and helping light to focus on the retina. And so we'll talk about that more when we get into the optics of the eye. So we have the sclera on the outside and the clear cornea over here as well. And these are both composed of the fibrous layer. The next layer over here is going to be the vascular layer in this kind of pinkish red color over here and here. And as the name suggests, it is rich with blood vessels. And they're going to be different regions. There's going to be the choroid over here. The choroid is going to be kind of deep to the sclera, and it's going to be rich with blood vessels, and it's going to supply the nutrients and oxygen that the eye needs to be able to function properly. The other structure over here is called the ciliary body. You may see it almost looks like flower petals going across the surface of the eye here. And it will actually extend up here. The ciliary body is important because it's going to contain ciliary muscles. And these muscles are going to attach to the lens using the ciliary zonules. or what are also called suspensory ligaments. And these ciliary zonules are going to attach the ciliary body to the lens. The muscles that are located within the ciliary body are going to play an important role in stretching and relaxing the lens. And this will help us to focus light properly onto the retina so that we are able to see a clear image. So the ciliary bodies are going to help focus the light by changing the shape of the lens. The other structure we have here is the iris. The iris is the colorful part of our eye. And so, and it's going to have muscles inside of it that are going to allow for the pupil, the hole that is in the center of the iris, to expand and contract. The iris plays an important role for controlling the amount of light that enters the eye. So we want to make sure that we have just the right amount of light entering the eye. And it also is going to aid in some optics. It's going to help limit which rays of light enter the eye, and it will help focus the eye on objects that are located nearby. So we'll talk more about that again when we get into our section about the optics. So here you can see it a little bit closer. Here we have the vascular layer. This is going to be the choroid that is going to be rich with blood vessels over here. This is going to be the ciliary body here and over here. The petals are called ora strata. And the ciliary bodies are going to actually connect to the lens using the ciliary zonules, or these suspensory ligaments. Okay. Oh, I can't wait. So the iris does control the amount of light that enters the eye, and to be able to do this, we have two muscles within the iris. We have the sphincter pupillae and the dilator pupillae. Pupillae refers to pupil, so it's the sphincter and dilator of the pupil. Over here we have the sphincter muscle located here. It's going to be a circular muscle, and when it contracts, it's going to decrease the size of the pupil. And over here, this kind of radiating muscle out is going to be the dilator. When that contracts, it's going to increase the size of the pupil. Under parasympathetic stimulation, what will happen is the pupil will actually constrict, because the pupillary sphincter will contract and the size of the pupil will actually decrease. And then over here under sympathetic stimulation, what will happen is the dilator muscle will contract and it will dilate. Fun fact, you can learn a lot about what is going through a person's mind when they are thinking, and you can use the pupil to help you out. When a person is really lost in thought or trying to think about a particular subject really well, or do like a calculation in their mind, what will happen is their pupils will actually dilate, and the amount of dilation is going to be proportionate to the difficulty of the problem. So fun fact, that's a thing. Also, if if you have a crush on someone, and if you're looking at someone, your pupils will also dilate because you're getting that kind of sympathetic stimulation. And so you can tell if someone has a crush on you if their pupils dilate every time they look at you. So that's a thing. There used to be a compound called belladonna that people in France used to put in their eyes to dilate their pupils because they thought it made them look more attractive. Unfortunately, belladonna is poisonous and it caused a lot of blindness in addition to dilating the pupils. Okay. Yes, no, maybe so. Okay, fun facts, random things. Now let's talk about the inner layer of the eye. The inner layer of the eye, or the neural tunic, is going to be the deepest layer of the eye, and it's illustrated here in yellow. And this is going to contain things such as the retina. The retina is going to contain the photoreceptors that are responsible for detecting light and transmitting that light into an action potential. And it's also going to contain the optic nerve over here. The optic nerve will contain the axons that will move away from the retina and carry visual information into the central nervous system. The retina is actually composed of two layers. So if we're looking at the retina itself, I'll show you in a different diagram in just a moment. But it has a pigmented layer and a neural layer. The pigmented layer is going to have specialized melanocytes, which are really important because melanocytes are going to produce melanin, that pigment that is also associated with eye color and skin color and hair color. And this melanin is going to absorb excess light, and it's going to help prevent light from scattering in the eye. So this is going to ensure that we don't accidentally activate photoreceptors in our retina by having light bounce around in our eye. So it will absorb that excess light. There's also going to be the neural layer. The neural layer is going to contain photoreceptors and other types of neuron that are going to integrate and process visual information, and then send that information to the central nervous system for higher levels of integration. So here we're seeing those layers a little bit more closer up. Here we have the sclera, which is part of the fibrous layer, the choroid, which is part of the vascular layer, and very, very tiny, we have the pigmented layer and the neural layer of the neural retina, or of the inner layer. Here we can kind of zoom in a little bit further on the retina and see here, this is going to be the pigmented layer that's going to contain the melanocytes that are going to be rich in melanin, and that will prevent the scattering of light. So it will absorb excess light. And this layer over here is going to be the neural layer, and this will contain the photoreceptors that are responsible for detecting light. Let's look at that neural layer a little bit more detail and talk about the different receptors that are going to be in this retina. So over here, what we're going to have is we're going to have a layer of photoreceptors. Photoreceptors are going to have special proteins that are going to be associated with vitamins, specifically vitamin A. And these vitamins are capable of interacting with light and changing their structure. And when light hits them and these change their structure, the photoreceptors are going to become activated, and they will communicate with other neurons that are within the retina. Over here we have the photoreceptors. They're going to interact with the bipolar cells over here. So again, you can see this rare cell existing again in the nervous system within the retina of the eye. These bipolar cells are going to take information from those photoreceptors, integrate that information to some degree, and then send that information to the ganglion cells over here. The ganglion cells will again process that information and send that information to the central nervous system via its axons. I do want to remind you about some of that neural integration and kind of circuitry that we talked about in the past. Remember, the retina is going to have convergent neural circuitry. So here you can see many photoreceptors are connecting to one bipolar cell, and this bipolar cell is processing that information, that is being collected from those photoreceptors and determining whether or not it should fire. And then what will happen is those ganglion cells are also going to process that information and determine if it's an appropriate time to fire. Here you can see that this ganglion cell is connecting to multiple bipolar cells over here. So we can see that convergent neural circuitry occurring here. In addition to having those 3 layers of cells, we're going to have additional cells that are going to act as interneurons and continue with this neural integration and neural processing. There is going to be the amacrine cells. The amacrine cells are going to lead to neural processing between the different ganglion cells. So it'll connect multiple ganglion cells and allow for them to communicate and discuss with each other for what is the appropriate action they should take in response to the stimuli that they are receiving. There are also going to be horizontal cells located here. Horizontal cells are going to connect between different photoreceptors and allow for that integration and communication there. So even before we get into the brain, the retina is doing a lot of neural integration and processing of that visual information. And then eventually that information is going to be sent on to the central nervous system where it will be continued to be processed in the higher centers of the brain. So very complex, very cool, very interesting. So here you can see those cells a little bit better. Here we have the photoreceptors. We have the horizontal cells over here, allowing for integration between those photoreceptors. The bipolar cells located here, and the ganglion cells located here. And we have the amacrine cells that is allowing for integration here. So cool stuff. All right. Here is what the retina would look like under a microscope. So under a light microscope. Here you can see the choroid over here. This is going to be the area that is going to be rich with blood vessels. And here we're going to have the pigmented layer. These pigmented layer are going to have the retina, the melanin in the retina that is preventing the scattering of light. Here we see the nuclei of the rods and cones, which are the photoreceptors. These are going to be the nuclei of the bipolar cells. And here we have the nuclei of the ganglion cells. And the ganglion cells' axons will be located here, and they will eventually become the optic nerve. Okay. There are 2 major types of photoreceptors. There are going to be rods and cones. They are named based on their structure here and here. So the rods are going to see light in grayscale. So it'll see in black and white. And they are best used in low light vision. So when we are outside in the dark, we're going to be using our rods to be able to see in the dark. Unfortunately, rods have a low acuity, meaning the image isn't very clear and it's a bit blurry. So that's why it's sometimes difficult to see in the dark. We also have cones. cones are going to be able to detect light. There's going to be different cone. There are 3 different types of cones that are going to detect light. So it's going to be red, green, and blue. Those will be the types of cones. And depending on how the cones get stimulated, it'll lead to all of the different colors that we are capable of seeing. Cones are actually going to have a high visual acuity, and so they're going to create a very crisp and clear image, unlike the rods. Before we leave our discussion of the retina, I do want to talk about special regions that are found in the retina. And when we look at the retina in the back, there is the most posterior part of the retina, and this area is going to have kind of a yellowish color, and that's why we call it the macula lutea over here. And the center of the macula lutea, the center of the macula lutea is going to have the fovea centralis, the central kind of location. The fovea centralis is really important because it has a high number of cones. And when light is going to focus on the retina, light is going to kind of focus on the fovea centralis. So this will be the focal point of our eyes. And then another structure that we see over here is we see the optic disc. The optic disc is going to be a collection of the axons from the ganglion cells. And that will be the first part of the optic nerve. In that region, there is no photoreceptors. So each of our eyes actually has a blind spot. You can actually play around and close one eye and try to find your blind spot by moving your thumb around. Eventually what will happen is if you find the right spot, you won't be able to see your thumb. Rather, the information that you'll see is the images behind where your thumb is located. The reason why this occurs is that vision is not only about information that is coming into the brain, but also what the brain is contributing as well. Sensation and perception is a two-way street. So even though you have a blind spot in each eye, your brain is compensating for it. So most of the time you don't even notice it until you actually look for it. So there is the macula lutea, the yellow spot in the back of the eye, the fovea centralis, which is in the center of it, and that's going to be the focal point of the light, and the optic disc, which is going to lack photoreceptors, and it's going to be kind of the starting point of the optic nerve. Okay. Before we move on to the physiology of the eye and how vision works, we're going to need to talk about the different chambers and fluids of the eye. The eye is divided into segments and chambers because the eye is a hollow structure and there are special regions within the hollow structure. And these chambers are going to be filled with fluid. And there are 2 types of fluid that we'll discuss: vitreous fluid and or vitreous humor and aqueous humor. So let's talk about these chambers. The first thing I'd like to discuss is going to be the segments. The segments of the eye are going to be divided by the location of the lens. The anterior segment is going to be anything that is in front of the lens, so between the lens and the cornea. So that will be the anterior segment of the eye, that hollow space. And over here, anything behind the lens, from the lens to the retina itself, that's going to be the posterior segment here, that hollow space. So the anterior segment and the posterior segment. The anterior segment can be further subdivided into chambers, and that is divided by the iris. In front of the iris, we have the In front of the iris, we're going to have the anterior chamber. And then over here between the lens and the iris, we have the posterior chamber. Those two chambers can combine to form the anterior segment. These segments of the eye are going to be filled with different types of humors or fluids. Humor is an old Greek word for the fluids of the body, and so we still kind of use that in anatomy. And we're going to talk about the vitreous humor and the aqueous humor. The vitreous humor is going to be found in the posterior segment, and it's going to be a jelly-like substance that is really good at holding on to water. About 98% of this vitreous humor is going to be water, but it's going to be like a jello-like substance. This vitreous humor actually forms in utero, in the embryo. And what will happen is this will actually last throughout the lifetime of an individual. Oops. So what are the functions of the vitreous humor? It's going to transmit light, and so it's going to allow light to pass through its structure. It's also going to help support the lens. It's going to help support the lens on the posterior side, kind of helping hold it up. And it also is going to connect the neural retina and kind of push the neural retina against the pigmented layer. If you're doing a dissection of the eye and you remove the vitreous humor, the neural layer of the retina will come off and slide off relatively easy. So this vitreous humor is going to kind of push the those two sections together and hold them in place. It also helps maintain intraocular pressure, so it's going to help keep the shape of the eye and hold it in place. We also have the aqueous humor. The aqueous humor is going to be in the anterior segment of the eye, and this is going to actually be renewed continuously. And the functions of the aqueous humor is to maintain intraocular pressure, mainly within the cornea, and it's going to supply oxygen and nutrients to the cornea. Because the cornea is avascular, it doesn't have any blood vessels, so it can't feed itself. So the aqueous humor is going to feed it instead. If we did have the ability to do the cow eye dissection, one of the interesting features of the cow eye dissection is that when you cut into the eye, sometimes the aqueous humor can squirt out because of that pressure. So be careful of that if you ever do a eye dissection. I'm not going to make you know the individual steps of this feature, but I do want you to know that the aqueous humor continues to be regenerated and flow through the eye. It's going to be made by the ciliary body, and it will flow behind the iris, over the iris, and then in front, filling this chamber over here. And eventually it will drain away into the scleral venous sinus over there. So those are the chambers and fluids that I'd like you to be familiar with. The last thing I'd like to talk about in this section on the anatomy of the eye is the lens. The lens is a transparent biconcave structure, meaning that it kind of curves out. And the lens plays a really important role in focusing light. The cornea, in reality, bends the most light, so it's going to refract the light the most. But the lens is really important because it fine-tunes that refraction and helps focus the light onto the back of the retina. And it can do this because it's attached to the ciliary muscles, and the ciliary muscles will stretch and relax the lens, so the lens can actually flatten and bulge to control where that light hits. In our next section, we'll talk about the eye as an optical device. And so we're going to start looking at the physiology of the eye and how everything kind of comes together to allow us to see. All right, have a wonderful day and be good to yourself. And I will see you in the next video. Bye-bye.